Good afternoon, everyone. The title of discussion today is Digesting Gastroenterology. So we're exploring the use of AI, artificial intelligence, machine learning, and software development in gastroenterology and hepatology. Now, the topics that we're going to discuss today, I'm going to provide a background on artificial intelligence and its history. I will talk a bit about software development, and we'll also talk about some case studies that we have in GI and the application of machine learning and AI in GI analysis, whether it's esophageal motility, ultrasound, gene expression, and etc. Okay. Now, why is it combining medical knowledge and technology important? It enhances diagnostics and treatment, allows personalized medicine, efficient data management, research and innovation, remote patient monitoring, global accessibility, and data-driven decisions, which allow patient empowerment. Now, artificial intelligence. So AI itself is the broader concept of enabling a machine or system to sense, to reason, to act, or adapt like a human being. Now, machine learning is an application of AI that allows machines to extract knowledge from data and learn it from it automatically, okay? So machine learning is a subset that learns from data without being explicitly programmed. It has applications in image and speech recognition, processing, medical diagnosis, prognosis, and recommendation systems, okay? Now, machine learning itself, it branches out to different sub-branches: supervised, unsupervised, and reinforcement. So we have classification in the supervised, clustering onto unsupervised, used regularly in genomics, and reinforcement learning and decision-making. Now, if you have an example of a traditional machine learning or a shallow learner compared to a deep learner, in the shallow learning case, the user or the expert has to extract features and then feed them into a classifier to produce an outcome. Whereas in a deep learner, the feature extraction and learning and classification is done automatically by the deep learner. Now we could have an example, with a precancerous lesion in the upper GI endoscopy in A. So the physician would look at the image, we would just do some inferences in his brain and produce a diagnosis. Whether machine learning, the expert should extract some features from the data and then feed them into a classifier to produce the outcome. Where there's a deep learner, it can extract the features and do the classification all by itself. Okay, and there are a few misconceptions about AI. Now, but they all share a common thread. They don't work in AI themselves. So they seem to actually confuse the two terms, and that is natural human intelligence versus artificial intelligence. Now, quite complex. A lot of human jobs require complex decision-making, creativity, empathy, or nuanced understanding, which AI actually struggles with. Another misconception is AI is infallible and is objective, but AI systems are trained on data, and if the data is biased or flawed, AI can inherit those biases. AI has common sense and reasoning abilities. I mean, AI really lacks human reasoning. Misconception number 5, AI will have emotions and consciousness. I mean, while AI can simulate emotions, but these simulations are not genuine emotions. And last but not least, AI is a threat to humanity and will take over the world. I mean, my AI becoming a hostile force is probably more science fiction than reality. But there are also valid concerns about how AI could be misused, or ethical considerations are crucial in its development. So what is intelligence? Okay, so what capabilities should a machine have for us to call it intelligent? So there's this test, they call it the Turing test. It says if the human cannot tell whether the responses from the other side of the wall comes from a human or computer, then the computer is intelligent, okay? Now, do we seek performance or do we seek human-like behavior, okay? So can you get a computer to do something that you don't know how to do? Or what about creativity? So in order for a computer to act intelligently, it should think humanly, it should think rationally, and it should act rationally, okay? And the story of AI is nothing new. It goes back to the 1950s, where the term artificial intelligence was adopted. But because due to the lack of computational power, they had this period, they call it the AI winter in the 1980s. But with the advent of new technology and processing power, or different learning methods for neural networks, now we see a lot of interest now back in AI, in especially deep learning. which is based on artificial neural networks, convolutional or recurrent. So what is AI, okay? So AI can be broadly defined as a technology that can learn and produce an intelligent behavior. Now we have, let's say we have a CT image of a liver with cirrhosis, okay? So we have an AI process. So we expect the AI to learn how to classify this disease, right? This can be like in also like say language translation. So if we think of it as incoming impulses passed through one neuron, okay, to the next, generating an output. So AI is essentially a mathematical function. So we can connect as many of these neurons as needed, resulting what is called a neural network, right? So the more layers we add, the deeper it becomes. That's why they call it deep learning methods, okay? Now, going back to the cirrhosis image, now, if you have like, if you want your AI system to learn how to classify different diseases, so you have to train the AI on the different patterns. So first you run the AI or the neural network on a sample. You do it then on a second sample, so you know the sample, and you repeat this for all the groups. And the goal is, once you pass all these samples, if you have a new patient, when you CT image, you expect the AI to produce the right outcome, okay? I mean, deep learning itself has a lot of sub-branches. What you see in the literature right now is a lot of CNNs or convolutional neural networks, and they are mainly designed for processing images and videos. They also have recurrent neural networks, which are suitable for analyzing time series and natural language and speech processing. Then we have generative adversarial networks, which have two neural networks competing with each other. They're used for image generation and data augmentation. We have autoencoders. They're used for unsupervised learning and dimensionality reduction. And a very popular one, transformers, they are used for natural language processing. So they have this mechanism, it's called self-attention. It captures contextual relationships between different words or tokens. I mean, one popular one is actually ChatGPT, so you guys probably know. So they combine transformers with reinforcement learning. So every time you use it and you provide your input, you're helping the company. Now, we have graph neural networks. They represent like, molecular structures or social networks. We have neuroevolution. We combine neural networks with genetic algorithms. We have meta learning, where we don't have actually a lot of labeled data. We have few shot learning, when we have only just a single example of a disease. And last but not least, we have explainable AI. So it's not specific neural network architecture, but actually it allows humans to understand what goes on within the neural network. And there are other neural networks that we're not going to talk about. So AI and machine learning, gastroenterology and hepatology. So it has been used in the identification of pre-malignant and malignant lesions. It has been used for the detection of lesions such as polyps, or small bowel bleeding lesions on capsule endoscopy, or pancreatic cystic lesions. It has been used in predicting disease prognosis or treatment response. Also, it has been used in IBD, determining the percentage of patients with inflammatory bowel disease. So the pipeline would look something like this, the AI-assisted endoscopy and radiology. So you would have data from the patient, where there is a multimodal, where there's endoscopy, radiology, CT, MRI, or pathology. And then you would choose your classifier, whether you want a traditional machine learning or an AI, a neural network or a deep learner. And the outcomes would be like prediction of prognosis, evaluation of surgery, treatment plan, classification, or diagnosis, okay? Now, but AI has often different uses as well for the early detection of prognosis and treatment of GI diseases. It can be used for detecting anomalies. It can be used for personalized treatment, recommend tailored treatment plans and optimizing outcomes. It can be used for predictive modeling, for predicting disease progression and treatment. It can be used in pathology analysis, treatment optimization, medication management, like enhancing low quality medical images, natural language processing, like, we want to extract valuable information from clinical notes and reports, telemedicine, which actually, chat is a chatbot. Actually, ChatGPT is also a chatbot that we can use it actually for medical data, prognosis, virtual consultations, quality improvement, and AI-driven artificial simulations, okay? Now, software development. So another important thing is like, in order to bring AI and machine learning methods to life, you have to have software so the users can use it, right? So, but development of software, it requires design, coding, testing, deployment, and maintenance. So it's equally important. So the intersection of machine learning, AI, and software actually will promise revolutionary breakthroughs across industries. And there are actually free tools, like Google uses Python, like, you know, they're free for visualizations, for training your networks and coding. I mean, software itself in GI has been used a lot for data management in electronic health records like Epic or MyChart, in telemedicine, procedure planning and simulations, image analysis, decision support systems, and patient engagement apps. And there are many other uses of software in GI, whether it's like medical image analysis, or training in simulators, or real-time monitoring, or patient portal integration. Now, as I was just trying to get to, I'm going to discuss some case studies to see like some applications of AI in GI and also in liver disease, okay? Now, the first case study is studying the Phreno-Esophageal fat pad, okay, in healthy and patients with achalasia, okay? Now, achalasia is a motility disorder, which is like failure of the lower esophageal sphincter to relax. The basic pathology of achalasia involves the loss of nerve cells in the myenteric plexus, leading to difficulty swallowing. So diagnosis involves various tests such as barium swallow, manometry, endoscopy. The hiatus itself is formed by the right cruise of the diaphragm, okay? And under resting conditions, it's physically superimposed on the top half of the smooth muscle LES in normal subjects. So the formation of a small sliding hernia, okay, with swallows implies that the lower esophageal sphincter slides from its location in the abdomen and the hiatus to the thorax and back during swallowing, okay? So the fat actually acts kind of like a lubricant, okay? So the fat in the esophagus, the hiatus, like acts as a lubricant. So it allows sliding between the LES and the crural diaphragm in normal subjects. But what we observed, actually, this was published like a week ago, in the achalasia esophagus subjects, there is a loss of fat content in these subjects, okay? So this sliding becomes rigid. So, but how do we prove this, right? So first we have to acquire CT images. We have to locate the different regions of hiatus, muscle, and fat. Then we have to use machine learning to compare the two groups, okay? So we obtained 15 abdominal CTs of normal and achalasia patients. And we did some grayscale metric analysis, whether you do on the intensity or the Hounsfield, and we developed a custom-built software to do this. The upper three panels are the hiatus of the three normal subjects. You see the darker regions. And you see the patient's achalasia, so it looks like more muscle, no dark regions there. So how do we prove there is more fat in the normal subjects compared to patients? So we developed this software, it's called SecCruise. So in SecCruise, the user can load, let's say, a CT image, and then they can crop the area of interest, which includes the hiatus region. It contains fat and it contains muscle. Then the program will ask the user to draw the esophageal hiatus region. So you can do this by clicking around the region of interest. And once you do this, we get some regional intensity. And then you do the same thing for the muscle. And then you will do the same thing for fat. In this way, we collect intensity measurements of different regions to make those comparisons. And you can carry this process for all the subjects in your database, okay? And once you do that, the program automatically will spit out two things. One is the histogram distribution of the intensity images of the fat of the muscle. And we are interested to see the overlap between the fat and muscle regions and the hiatus. And it also gives you a segmentation of your CT image. The output you can get is a quantitative output. Like one of the good features is the overlap of the hiatus region with muscle and overlap with fat. So you can repeat this for 50 normals. The solid lines are each individual normal. Then you can use machine learning to get the mean of each region for the normals. You can do this for the patients. And then once you build the mean, you can compare the two groups. That's 15 normals in the upper panel and 15 in the achalasia. So you can see the hiatus region intensity is in normal, is closer distant-wise and has more overlap with the fat area.
What's the unit of the x-axis and the y-axis?
That's a grayscale intensity, okay? We're not using Hounsfield. So the color mapping is different, okay? And the hiatus region intensities in patients are closer to muscle. So they're correlated. They're both color maps, okay? You can go either way. So that's how we proved it. Now, the second case I want to is the use of AI in esophageal motility in functional dysphagia. Now, in order to understand functional dysphagia, we have to understand peristalsis. So peristalsis comprises of an initial relaxation followed by contraction. So it's a contraction and the distension. Now, high-resolution manometry is the primary method used to evaluate motor function. So the problem with high-resolution manometry that we actually use in our clinics, it only measures the contraction phase of the peristalsis. I see the yellow regions. What about the distension? How do we measure that, right? So now there's this consensus, I call it the Chicago classification, 4.0 right now, which actually classifies different esophageal motility diseases based on their contractile behavior. But what the Chicago classification fails to detect is like, what if the pressure has distension abnormalities, not contractile, right? Now, because we talked about peristalsis is distension plus contraction. So what if the problem is distension and not contraction? And here comes functional dysphagia, okay? So functional dysphagia right now is a diagnosis of exclusion. These subjects are classified as normal based on the Chicago classification. Nobody really knows the mechanism. They previously thought it's related to visceral hypersensitivity, inappropriate pain perception, or maybe abnormal contraction abnormalities. But our studies show, and they point to a new direction, that actually these patients have abnormal relaxation during peristalsis, okay? So this would have never been detected by CC, the Chicago classification, because they use only pressure. So, and functional dysphagia actually is common, like goes back to 1987, like 47% of the patients who came to Katzenkanstel, like, they were classified as normal, but they had dysphagia symptoms. 2008, work of Pandolfin and Karelas, 25.6%. Our own motility patients that come, 38% of them, they're based on Chicago, they're normal, but they're complaining of dysphagia. If you look at their pressure manometry, they look pretty normal, right? So what's wrong with them? Are these patients paranoid? Or are the features we're using inadequate for detecting this kind of disease, right? So going back to peristalsis, like if you think about like a bolus as a rock and the contraction as a bulldozer, so the highway has to distend in front of the bolus for the bolus to go nicely smoothly into the lower sphincter, right? So what we see in functional dysphagia patients, interestingly enough, they have a narrowing of the esophageal region in front of the bolus, okay? So there has to be a mechanism to measure distension because their contraction patterns looks normal. So the first question is, now that we know functional dysphagia is affected by abnormal distension, how do we calculate distension, okay? The second question is, what features do we use to train, which classifier do we use? Do we use shallow or deep learners? And once we have this system, how do we make it available for other GI clinicians to use it? So actually, when you do a manometry in your clinic, you have two sets of data sets. Once you have the pressure, the 36 pressures, but you also have impedance, okay? So how do we convert impedance to distension? So we can, if you see the ultrasound of, let's say, of a swallow from 1 to 8, you can see the dilation of the esophageal at one level is actually correlated with the drop in impedance, the orange curve. So actually, you can use laws from physics, which tells us that impedance actually is inversely proportional to cross-section area. And you can measure distension, with a bit of modification. So the second question would becomes what type of classifier to use, okay? So as we discussed, we can use traditional machine learners or deep ones, okay? Deep learners, they require a lot of training data, okay? I see a lot of hype in deep learning, and I've rejected like many papers on deep learning because they just lack data. So where you can use like simple machine learning, shallow learners, why you would do complex stuff, right? So here we had 30 normals and 30 patients. We went the machine learning, the shallow learning route. So we had to extract some features. So some features that we use is T1 is the time from the pharyngeal opening to the onset of swallow peak distension, and T2, the time difference between the onset of swallow to peak contraction. You can also use the peak luminal distension or peak luminal contraction, and some other features, the area under the curve of contraction and area under the curve of distension, right? And then you can train different classifiers, logistic regression, support vector machines, random forest decision trees to get results. And we segmented the esophagus into 4 segments, and just using distension alone actually clearly classifies the functional dysphagia from the normal patients, okay? So you measure the accuracy, precision, or recall and compare the different algorithms, okay? And actually, once you build the distension profiles of these patients, you see, although the contraction looks normal, if you see the distension, the bolus, it's a narrowing in the esophagus as the bolus is going down the length of the esophagus. And so how do we make this system that developed available for other people to use? So we came up with this new software. It gives you the visualization of both pressure, impedance, and distension. It allows different visualization of distension, which is a primary problem with functional dysphagia patients. And so the clinician can see the distension phase plus the contraction that we've seen before, okay? And we also made it available in mobile portable phone. So you can just carry your tablet or mobile phone and do the analysis. The case study is ultrasound analysis. So another modality that actually they're commonly used in esophageal manometry, or let's say one of these EOE, the eosinophilic esophagitis. I mean, we want to learn about the thickness of the layer of the esophagus, okay? So that's why we use esophageal ultrasound, okay? Now, the problem with ultrasound itself, there are no available systems to analyze it, okay? They're long videos, and if you want to, it will take days and days to do an analysis. So we came with this solution. We converting the video to a sequence of frames, then choose 6 in different angles, and then analyze the image semi-automatically by the user clicking on the motion modes across the frames. So this segmentation will take, say, under 15 minutes. And then you can get information as the muscle area, the lumen area, or the thickness across the length of the video. And then actually you can finally track your segmentations and see, like in vivo, the CSA of the different swallows, okay? But think about it this way. I want you to think about the stuff that you guys are doing too. This can be applicable anywhere, okay? Now, you can also use machine learning or AI creating deformable shape models of the human organs. So statistical shape models are used to represent and analyze variability of shapes. So machine learning can be used to learn the distribution of shapes and capture both the mean and variations around the mean. This is actually the meat slicing of the human visible project. And you can actually segment out the lungs, the bones, and this is actually the sagittal cut of the volume. And you do and then you can do segmentation to segment out the lungs, the thorax, the bones, and you can build a beautiful segmentation of the thorax. And then you can use machine learning to build deformed models of the lungs. Actually, this is actual lung during respiration, okay? So this is a computational model ready for, model analysis. And you can actually attach electrodes and put the lungs in the thorax and do this computations. So case number 5 is AI for predicting phenotyping on electronic health data. So before I get on, I say genotype. When I say genotype, we are referring to an individual's genetic makeup, whereas phenotype is the interaction between genotype and environmental factors. So what's the goal here? The goal here is precision medicine, okay? So let's say we have genetic, clinical, and environmental data, and we have state-of-the-art, let's say AI and machine learning, is coming up with predictive model for disease susceptibility and treatment response. So let's say if you have a new subject and you have the genetic, clinical, and environmental data, you come up with a personalized treatment, okay? That's the goal. Now, let's say you have an electronic health record of a subject with heart problems, and you can track this disease progression with time. So you can build a history, let's say, of heart attack, and the symptoms they're having. You can combine this with demographic, diagnosis, lab results, vitals, and medication. And you can actually do a predictive modeling of what are the chances of this subject having a heart attack in the next 5 years, okay? So the questions could be explanatory, predictive, or casual. Explanatory is who really had a heart attack and when? Was patient, let's say, on different doses of warfarin? Was the doses stable? Or could be predictive? Who will have a heart attack in the next year? Who will have a heart attack if they take the drug? Or could be casual. How much will the patient reduce the risk of heart attack if he stops smoking? Okay. Now, case number 6 is artificial intelligence for gene expression and regulation. I think you guys probably are more. This is more micro and for gene expression and regulation. Okay. Now, what's the central dogma of biology? Genes act to create phenotypes through a flow of information from DNA to RNA to proteins, to interactions among proteins, and ultimately phenotypes. And the collection of individual phenotypes will be a population, okay? And the DNA has 4 bases: the cytosine, thymine, adenine, and guanine. But how do you analyze large amount of genes, thousands and thousands of genes? So the tool is called microarrays, okay? So it allows us to detect the expression of thousands of genes at the same time, okay? Now, gene expression is the process which synthesizes a protein by using the information in the gene, whereas gene regulation is the process of controlling the rate and the manner of gene expression. So traditionally, there have been different, like 3 different approaches for gene expression. Has been identifying genes and biological pathways differentially expressing disease. It could be network-based, which identifies networks of genes differentially regulated, or it could be program-based. You identify disease-related differences in program progression, and you find which genes are upregulated, downregulated, or remain unchanged. So the questions are real traits dependent upon levels of gene expression? What is responsible for trait levels? The variation in gene expression or variation in the type of protein that is produced by the gene? And is your fate associated with genes that are on or off at some point in your disease? Or is a candidate's drug fate associated with a gene that is on or off in early trials? That's, for example, gene Q325. If it shuts down, then we have like liver failure, I mean, which can save the drug companies like, millions of dollars. And so recently I sat down with one of the faculty here, Dr. Dar, and he showed me this interesting paper on the use of actually a change of diet, how a simple change of diet can affect the progression of liver disease, which I found interesting. So here I'm going to just, and actually you can go download like sample data sets from the NCBI, like, if you want. So non-alcoholic steatohepatitis, like, is a type of a liver disease, which has inflammation, accumulation of fat in the liver, which can lead to liver damage and scarring, okay? Now, I'm gonna just propose a general pipeline that what can be used in gene expression using machine learning. So the first stage would be data collection and integration. That would be collecting gene expression profiles, then methylation patterns and transcription factor binding sites, and then also patient demographics, disease severity scores, and etc. Then the second stage would be feature engineering. That will involve selecting relevant genes, epigenetic regions, and features for further analysis. Then would be the building of gene regulatory networks. It can be done through graphical models or Bayesian networks to infer gene regulatory networks from the integrated data. Then we can select the features by doing dimensionality reduction. I've seen like a lot of papers that use principal component analysis or TNSE to retain essential information. Then we can use this data for predictive modeling to predict disease outcomes or predict the NASH progression or fibrosis stage. We can identify biomarkers and targets. We can actually score features by importance, and these features would serve as potential biomarkers for disease diagnosis and severity assessment, okay? And also, they can identify regulatory nodes as therapeutic targets. And then finally, we can interpret regulatory patterns to understand the contribution of each feature to model predictions, and finally validate these findings in the clinic, okay? That's like a general pipeline that we could use this for that. So an example, actually, we have this, the discovering patterns in gene expression profiles. Is from the paper exploring the metabolic and genetic control of gene expression in a genomic scale. It's from Science. So the goal was here to use DNA microarrays to study temporal gene expression of almost genes in the Saccharomyces cerevisiae during the metabolic shift from the fermentation to respiration. Now, once the supply of glucose exhausted, okay, the yeast fungus shifts from anaerobic fermentation of glucose to aerobic respiration of ethanol, okay? They call this process the diaxic shift. And this process is of considerable interest since it's accompanied by major changes in gene expression. So what they did, they measured expression levels at 7 time points during the diaxic shift. And they found out that this specific gene, the ACS1, appears to be strongly upregulated during the diaxic shift. And actually, they compared it with the other 6,400 genes, and the upregulation is still there. You can use different machine learning methods to actually cluster the profiles of these genes. You can use k-means clustering. or you can get the centers of these clusters, or you can use what you guys typically use, hierarchical clustering. I've seen a lot of papers use that. And so you combine the dendrogram with the clustergram. You can label these nodes. You can threshold them at different thresholds to study further the groups. You can zoom in the profiles. You can use different color maps to study these profiles. Or you can use other dimensional regression, such as principal component analysis or nonlinear principal analysis or factor analysis to get a different clustering compared to the histograms that you guys get, okay? And the second example is identifying overrepresented regulatory motives. This is from the paper Orchestrated Transcription of Key Pathways in Arabidopsis by the Circadian Clock. So the goal was to search for potential regulatory motives in a set of co-expressed genomic sequences by identifying overrepresented motives. So this, as you know, the circadian clock is a 24-hour cycle of the physiological process that's synchronized with the external day-night cycle. Most of the work of the circadian clock has been done on the Arabidopsis thalinia plant. So in this organism, the regulation of a series of genes that need to be turned on and off at a specific time of the day and night is accomplished by small regulatory sequences found upstream. And they call this regulatory motive the evening element, the AAA-TATCT element, which has been identified in the promoter region of the circadian clock-regulated genes that show peak expression in the evening. So how do you prove this? So they consider 3 sets of clock-regulated genes clustered according to the time of day when they were maximally expressed. That was 8 a.m. to 4 p.m., 4 p.m. to 12 p.m., and 12 p.m. to 8 a.m. Because they were interested in the evening genes, so set 2 was represented the target set, and set 1 and 3 were as the base sets. And after machine learning algorithms, they found out that the evening actually motive clearly stands out. But the problem with this motive findings, like there's a lot of redundancy, so you always have to filter out. So after repeated filtering using again machine learning algorithms, they found actually the first 2, the prominent ones, is just the evening motive itself and its reverse complement, and the other ones are just the variations. Okay. Now, how do you prove that your findings is statistically significant? So you can actually generate random sequences according to the nucleotide composition of the heterogeneous sequences. And you can see here in the figure, the E motive is clearly significantly larger than the control margins. So we can conclude that our findings are statistically significant, okay? Now, you can determine the presence of this evening motive among clock-regulated genes. So although we identified this motive, but expression peaks in the evening, it is not shared by all of evening genes, okay? Nor is it exclusive to this gene. So we can count the occurrences of this motive in the 3 sequences. And it appears that there's 9% of these genes in set 1, 40% in set 2, and 30% in set 3 have the E motive. Thus, not all genes in the evening set have the motive, but it's clearly enriched in this group. And you can also find the location and occurrence of this motive in the transcription start site. I'm not gonna discuss further. I just wanna so challenges in using AI. Okay, while everything looks very exciting, but there's still challenges, and one is data privacy and security. There are ethical considerations. There's regulatory compliance, data quality integrity, and always resistance to change, and many other kind of concerns. So regulatory landscape, then you have to deal with the FDA approvals, the compliance with the medical standards, and also patient safety is a priority. But the outlook on the future looks very bright, and the technology integration will enhance patient care, accelerate research, and improve healthcare accessibility. But this requires actually a collaboration between physicians, data scientists, and software developers to achieve this. So this is like a call for action. Thank you.
Great job. Thank you. Do you want to go to the Zoom view so we can see everybody? Sorry, Lars.
No, that's fine. I agree. So that was a pretty broad overview.